James McAvoy, and the title of the presentation is Understanding the Mechanism of lipopolysaccharide induced homolysis of whole blood. Yeah.
Well, hello, and thank you, Navi, for the introduction. As you said, we did our presentation over our research project, Understanding the Toxic Mechanism of Lipopolysaccharide. My name is Brendan Broders, and let's kick this off. All right, so let's start with a roadmap. First off, I'm going to give an introduction, then we're going to state a goal, we're going to talk about the methods, the results, and finally we're going to summarize. But let's meet our team first. My name's Brendan.
My name is Ashwin Mudaliar.
I'm Jordan.
My name is Benjamin.
Nathan.
All right, so lipopolysaccharide, what is it? It is a molecule found in gram-negative bacteria, specifically in the outer membrane as shown above. It is known to cause septic infections and ultimately septic shock, and it is an endotoxin or a toxin from within. Sepsis is deadly. This is why we kind of want to study it. And it is also costly at approximately 20 billion a year annually in the US. The only mechanism known right now is in response to the immune system. So what we wanted to do is we wanted to explore its interaction with the cell membranes of red blood cells and whole blood. All right, but first let's talk about these structures that we have in front of us. Figure one here is the lipopolysaccharide molecule. I want to draw everybody's attention to this area right here. Lots of non-polar, or they don't like water, hydrocarbon chains, and a sugar head group, which does like water, because you can see all the oxygens around it, head group. And then this molecule here is a glycerol phospholipid, as you see here. Similar to structure is the non-polar hydrocarbon tails and the polar head group. Now, this molecule here is found in our plasma membranes of almost all tissues and cells. So because of that, we kind of targeted this to our actual study, like the similarities between the molecules. But you guys might be asking, why do we care? Well, big picture is, if we can find out this mechanism or how it works, we can target an intervention or treatment that would reduce on mortality or death, and inpatient hospital stays, which would ultimately reduce that 20 billion a year spent in the US.
So, what's the primary goal of this this research we did? So, we were trying to look at when whole blood is subjected to LPS, what would happen? Would there be a decrease in cell number? Would there be, would it be due to a membrane interaction, as Brendan mentioned, with the polar groups and the non-polar groups? So that's kind of what the overall big picture we were kind of looking at. So, in order to do this, we had a control sample, which was just whole blood and saline, physiological pH saline, which I will refer to later on. We will refer to it later on as LPS negative. And then we had an experimental sample, which consists of LPS, red blood cells, and saline, which we'll refer to as LPS positive. So we took those, we mixed them up, and through a series of dilutions to get countable cell numbers. And we vortexed these in Eppendorf tubes gently in order to avoid lysing or breaking apart the red blood cells. We incubated them for 10 minutes at 36 to 38 degrees Celsius, which is right around human body temperature. We took 10 microliters, which is a very small amount, and put them on a hemocytometer and put those under the microscope to count the number of cells, and I'll get to how a hemocytometer works in the next slide. And then we took pictures. They were right after the 10 minutes, which we call the initial, 30 minutes, 60 minutes, and 90 minutes, and finally over 24-hour span, which we'll have in the upcoming slides that you can kind of see. So here's a quick summary of how a hemocytometer works. It consists of a mounting support and a cover glass, and underneath you have a filling notch and the counting chamber, which are two important concepts of it. So you take your pipette, you put it underneath the filling notch, and you just inject it in there, and the capillary action will take it your blood and your saline all across it, And then you'll be able to see the covering, the counting chamber here that has a grid on it, and from there you can count the number of cells in each grid, and this will give you it's kind of like a manual way to count cells.
So as we get into the pictures that we took with the hematometer using a motocam attached to our light microscope, here we see cells with LPS. On the left here, this is initial time. So we counted 33 cells compared to the right picture. There are 18 cells. This is after 24 hours. So visually, there is a decrease in the total number of cells, and as you can see, there are a little bit of background debris. For reference, this is a red blood cell, and things like this, we consider background debris. So then when we move to the cells without LPS, visually there are more cells compared to the initial time and 24 hours. For this trial, we had 112 cells that we were working with, and after 24 hours, there were 100 cells left over. So as we get into morphology, or those of you who don't know, this is just how they look. So again, this is just a regular blood cell. Some things that we wanted to highlight on are circled in red. These are cell debris. There's no definitive thing on what we can tell with this and what they are exactly. So we assume that these are cellular components, cells that have lysosed or just kind of broke apart and congregated with each other. This may even be the LPS molecules kind of sticking with each other, since they are in a polar environment, they like to stick with each other. This picture is whole blood with LPS at time 0 as soon as we mix them. And then this is the same sample, hour and a half later. As you can see, there's increased cell debris in the background. Notice in the top middle here, there is a cell that we assume to be undergoing lysis. Again, we don't know for sure, but after many trials, we assume that this is what's happening. In the bottom left there, we do see some more debris. And again, this may just be cellular components leaking out of cells, LPS molecules sticking with each other, or things of that nature. And then after 24 hours, cells with LPS, we see kind of the same trend here. More background debris. This middle circle, it looks kind of like cells stacked on top of each other. That may just be cells losing their pigment, or just undergoing lysing, or cell breakdown of that. And then we get into the cells without LPS, we refer to as negative control. After 24 hours, we don't see much difference in morphology or background debris, but this is just normative, just regular saline. Obviously, you will have a couple cells that look different. It is normal within the body for blood to regenerate and kind of die off, so that is normal to see.
So Jordan gave us the visual representation of this data, but how can we look at this in a quantitative way? In the blue, we can see initially at time of 0, we do reflect 100% of red blood cells present. This is going to be the same for our negative and positive LPS as well. But you can kind of observe here on the 24-hour count that there was a reduction with our red blood cells initially within the body. But for 24 hours with induced LPS, there is quite a significant reduction here that we can see. It should be noted that this is off of 5 samples in total. So with our experiment, we did do several trials, but with the data that's represented, it's going to be a totality of 5 here. With our standard deviation lines here, we can see with positive LPS in green, that's going to be the variation in the counts of red blood cells. So every time we loaded up the hematometer, we may have not had the exact amount of red blood cells every time. So that's why we have those arrow bars present there. So in complete count, Jordan showed you on the actual hematometer of how those red blood cells look. Out of all 5 experiments, we did see in our control group a total reduction of 3.45% of red blood cells. That would be naturally in our body without LPS being induced. But with the inducement of LPS, we can see in green here a total reduction of 31.5%. If that's happening in our body, that is very bad. That is not gonna be something that you'll be able to survive essentially, because that reduction ends up being a total amount of 28.05%. We did come up with a p-value here that reflects as 0.013. So our data is pretty significant based off of our findings. If we're referring back to a p-value of 0.05. It should be noted too that our t-score that we received here was 3.46. But Ash will kind of dive in a little bit more on the significance of these findings.
Yeah, to summarize, to see the clinical picture, how these things work. So for example, a patient comes to me with an infection, any kind of infection anywhere in the body, a bacterial infection. So the first thing prescribed is an antibiotic which kills the bacteria, right? And we said before that the LPS are present on the outer membrane of the gram-negative bacteria. So when these bacteria are dying, they break off the cell membrane. LPS are induced in the red blood cells, naturally in the body, which is when those things are induced, it depends on how bad the infection is. So normally our body is capable of taking those LPS out, but it takes a very long time, couple weeks to gradually throw them out. But as we see that there is a 28% of reduction of red blood cells within just 24 hours. So usually the old people who come up with a urinary tract infection comes to the ER, they do not have that much time, and with like getting older, their kidney functions and those stuff will also go down. So this affects significantly to those populations with a septic shock. And that being said, septic shock induces like a low blood pressure. The patients come up with a really low blood pressure and high heart rate and high fever and stuff like that. And we as clinicians and stuff, we just give them fluids which will increase their blood pressure, but they also have a risk of getting fluids filled up in their lungs, which we don't want. So eventually they could be transferred to the ICU, getting a dopamine infusion and stuff like that, which is like getting more worse. So we are just treating them symptomatically. That being said, we are also, for further to expanding this research, we are also doing couple research about the cells that they are dying because of the LPS, and tagging the LPS with immunofluorescent to find out the exact mechanism how it works. If we are able to figure out the exact mechanism, we can stop the mechanism somewhere in the